Hello, IB Biology students. This is Ms. Sheely here with your first installment of video lecture notes. Remember, your choice is to either read their textbook and take notes or watch the video lecture and take notes. So make sure that when you're taking notes, you pause the video, you write down anything that you feel is important, but also make sure that you record any questions that you may have, whether they be summary questions to help you reflect when you're studying and keep you on task, or they're questions of confusion so that we can go over them in class when we meet again. So the first thing we need to talk about is this idea of vitalism and molecular biology in particular. So we know that living and non-living things are different, but once upon a time, there was a theory of vitalism, and that was that living things are made of organic chemicals, we know, but that these organic chemicals can only be produced in living things because there was a vital force needed. So something inside of our bodies was needed to make those organic compounds. This was falsified by a series of discoveries that we're going to look into in the next minute. All right, so we're going to watch this video clip about scientist Frederick Wohler. And in 1828, he synthesized urea, which was really the starting point for falsifying this idea of vitalism.
Our next great discovery occurred in the 19th century, when many chemists believed that organic substances from organisms or living things were somehow different from inorganic substances from non-living things. But that was about to change. In 1828, Friedrich Wohler was working in his lab when something caught his eye. Wohler had placed two inorganic chemicals in a beaker, potassium cyanate and ammonium sulfate. Now when he looked at the beaker, it contained a gram's worth of small, white, needle-shaped crystals. What made this remarkable was that Wohler thought he'd seen these exact same crystals once before, but with an important difference. Those crystals had been organic. He had crystallized them while studying the chemistry of various substances found in urine. To make sure he wasn't mistaken, Wohler analyzed the new crystals. There was no mistake. These crystals were the same as those he had isolated before.
He had made urea, which was something that had come out of a living thing. He had made it out of inorganic substances. Later he said, in a personal letter, not in the paper he wrote about it, that I have made urea without a kidney. And he knew what he had done.
Meet Roald Hoffmann, winner of the 1981 Nobel Prize in Chemistry for developing a theory to explain organic chemical reactions. So why is this discovery of artificially making urea, why is that a great discovery?
You know, there comes a time when you need a discovery, and it's sometimes a single one, to cross a border, to break down a wall. This is what this discovery was. It's not that it was so important in and of itself, but at the time that it came, this simple making of urea out of two inorganic chemicals, when it came, it caught people's attention. The whole story of the discovery is about the underlying basis, the building blocks of all matter, organic and inorganic, being the same, atoms.
So this idea of vitalism and this scientist, Frederick Wohler, and his synthesis of urea in 1828 led to the disbelief of the theory of vitalism. And it really is a great example of what we call the nature of science, how there is a belief and then you have an experiment that shows some differing opinion. And then we expand upon that and can change the course of science as we know it. So it is very exciting. So, of course, we still, to this day, have never discovered a vital force. So, the best answer for what causes or allows us to make these organic things is natural selection. Now, we, of course, believe that living and non-living matter are governed by the same chemical and physical forces. So, this is really important because it does explain the living processes in terms of our chemical substances, and that's why typically in a biology class we will take a reductionist approach where we will start with the very, very small and then build upon that. The discovery of the double helix was another great nature of science idea where scientists really started looking at the structure of molecules but through models, and we'll explore this again when we get to DNA structure in a bit in the year. So now we're going to talk about a particular element, we're going to talk about many elements. So there are many elements, of course, that occur frequently in living organisms and are used in metabolic processes. The four most common are carbon, hydrogen, oxygen, and nitrogen, but you can see here in this periodic table that there are a few more that are shaded, such as sodium, magnesium, iron, phosphorus, sulfur, things like that. Now, carbon is a particular importance to us. It is the 15th most abundant element, but there's a few other things about carbon that make it extremely special. So before we really dive into carbon, we do need to remember what a molecule is. And a molecule is a combination of two or more atoms that are typically held together by a bond. The bonds that we're looking at today are covalent bonds. And this is a type of bond where we share electrons equally between the atoms. Of course, if you remember from chemistry, ionic bonds is when there's an unequal sharing of electrons. Covalent bonds can be single bonds, double bonds, or even triple bonds. And we will look at some of these examples in an activity that we're going to do in class. So because carbon has these four valence electrons on the outside on its outer ring, it can form four covalent bonds. It can share its electrons with other elements and atoms. And that really makes it very special. This is more than most other atoms and elements combined. Many other elements that are commonly found in nature form much fewer bonds. Hydrogen only forms one, oxygen can form two, and nitrogen can form three. Here you can see the versatility of carbon. We can have just a hydrogen shell around the carbon, such as in methane. We can have a linear structure where you have a triple bond between two carbons and then a single hydrogen on each end. You can have another linear structure but with more hydrogens. You can have a ring structure or even a branched structure as well. So this really makes carbon very, very versatile. But also because it typically does form covalent bonds, it shares its electrons. Carbon molecules are very stable because covalent bonds are the strongest bonds. Some specific organic molecules that we're going to look at in the next slide are sugars and carbohydrates, and you can see that they typically form a ring structure, whether that be a pentagon ring called a pentose for sugar, or a hexagon ring called a hexose. Proteins, which are made up of amino acids, have a very generalized structure where you have this amine group, which is a nitrogen with two hydrogens on the left side, a central carbon with this R group on top, and then on the right side is the carboxyl group where you have a double bonded oxygen and an OH group off of the carbon. And then lipids can be they're typically a linear structure, but they can be saturated or unsaturated, and we'll talk about that in a minute. And you can draw your lipid completely filled out like this, or you can abbreviated and have however many chains you have represented by the N. And we'll talk about that some more in class. All right, so again, the first of our main classes of carbon compounds is going to be carbohydrates. So if we remember from baby bio in 9th grade, carbohydrates are made of 3 elements: a carbon, a hydrogen, and an oxygen. And this carbon, hydrogen, oxygen is in a 2-to-1 ratio, and so we call it a hydrate because for every carbon there are 2 hydrogens. So it is a hydrate. So that's where the hydrate in carbohydrate comes from. Lipids are the next class. All lipids are insoluble. We actually have 4 classes of lipids: fatty acids, steroids, waxes, triglycerides. Lipids can be, like I said in the last slide, saturated or unsaturated. Saturated lipids have only single bonds, so there's no kinks or bends to them, and they are also solid at room temperature. Unsaturated lipids have at least 1 double bond, although they can have more, and this will form a kink or a bend in the lipid structure. It will not be completely linear, and these lipids are liquid at room temperature, so these would be like your oils. Proteins are the third class of carbon compounds. They are chains of amino acids, and so they are varied in structure. There are 20 different amino acids, and they can be arranged in any combination. And depending on their combination is going to depend on the functionality of the protein itself. Proteins are made of carbon, hydrogen, and oxygen, just like carbohydrates, and also lipids, which I neglected to mention. But proteins also have that nitrogen from the amine group. Now, there are 2 amino acids that also contain sulfur, but because there's only 2 out of the 20 that contain sulfur, we don't typically say that proteins are made of sulfur as well. Proteins run a huge range of tasks in our body, but the biggest one is really controlling all of the chemical reactions in all of our cells and thus all of our bodies. And this is our group of proteins known as enzymes, and we'll study them in much more detail in the future. Our last class of carbon compounds is nucleic acids. Nucleic acids are chains of nucleotides. They are made of carbon, hydrogen, oxygen, nitrogen, and also phosphorus. There's 2 main types of nucleic acids. There's DNA and RNA, and these are the chemicals that are used to make our genes, and thus our genes are used to make proteins. So these are very closely related. The last concept that we're going to look at today is an idea called metabolism. And your metabolism is the web of all of your enzyme catalyzed reactions in a cell, which could be a single-celled organism, or in the entire organism itself. These are reactions that are sped up or happen faster in the presence of an enzyme. Metabolic pathways can be either a chain, which would be a linear process where you can think more of like an assembly line, where you have reaction A is going to give you product B, then product B will go into reaction C, and thus until we get to our final product. But you can also have a metabolic pathway that happens in a cycle. And in this case, this is a cyclic relationship, and your final product may be spit out along the way, or it may be kept within the cycle itself. So we have 2 main categories of our metabolic reactions. We have reactions that make bonds, and we have reactions that break bonds. So it doesn't matter which one you're talking about. All of these reactions require enzymes, which are our biological catalysts. So when we're making bonds, we call this a condensation reaction. We call it condensation because just like when you are going outside and you notice a buildup of water, called condensation. So condensation means water releasing. Another term for a condensation reaction is called an anabolic reaction. Again, we're making bonds, so we're building molecules. This idea of anabolic is the term that IB does prefer. So be comfortable with either way, but definitely be comfortable with seeing the anabolic. So anabolic reactions that are going to build molecules are going to need some type of energy and are going to release water. The other type of reaction is called a hydrolysis reaction, and this is where we're going to break bonds and thus break down our molecules. So if you actually break down the word hydrolysis, hydro means water, lysis means splitting. So hydrolysis literally means water splitting. We are going to split a molecule of water to break a bond, and a piece of each of the water, one hydrogen and one hydroxyl group, is going to go to each piece that has been broken apart. This hydrolysis reactions are also called catabolic reactions, and again, they are the ones where we're going to break down the molecules. These are typically like your digestion. All right, so I think these videos will show us some visuals of both the anabolic and catabolic relationship. All right, so here we have two different molecules. You can see an OH or hydroxyl group is highlighted on this first molecule, and just the hydrogen out of the hydroxyl group is highlighted on the others. I'm going to start the animation.
A condensation reaction joins two molecules together to form one larger molecule. An enzyme removes a hydroxyl group from one molecule and a hydrogen atom from another, then speeds the formation of a bond between the two molecules at their exposed sites. Typically, the discarded atoms join to form a molecule of water. Hydrolysis is a type of cleavage reaction. It's like condensation in reverse. An enzyme splits the molecule into two parts, then attaches a hydroxyl group and a hydrogen group from water to the exposed sites. The result is two smaller molecules. Condensation and hydrolysis are used to put together and break apart many biological molecules.
All right, so I hope that animation helped you. Do remember, though, that IB is really going to prefer this anabolic to the condensation, where we're building molecules, we're making things, we're making proteins. This will be really important when we're doing that. And catabolic for our hydrolysis reactions, where we're breaking down molecules. All right, guys, that is it for our first section of notes. I look forward to seeing you in class and discussing this and diving into it further. Have a great day.